Hello. Yeah, I just shortly going to present myself. I'm a toxicologist by training. I work for Douglas Connect, and my main task within Douglas Connect actually was the launch of our lab. We started in September, so we're pretty new, but this is also one of the projects in which I'm involved. It's a Marie Curie actually sponsored project, and the idea is to develop a workflow for AOP hypothesis generation, and here we actually develop more in-depth the use case of non-genotoxic-induced hepatocellular carcinoma. I start with a short introduction on cancer and that it can be a consequence on a multiple source of exposure, and yeah, interestingly also from chemicals. You have two types of chemicals which can induce cancer: genotoxicant and non-genotoxicants. The genotoxic carcinogens are inducing, they can cause actually changes quantitatively or qualitatively on the genetic material, whereas non-genotoxic carcinogens, they do not bind to the DNA and are not mutagenic, but they can yet cause cancer. Nowadays, regulatory to identify carcinogens, we need to carry out the traditional 2-year rodent carcinogenicity bioassay, which unfortunately costs quite a lot of number of animals, around 800 animals per compound tested, so alternatives are sought. In vitro, what's available nowadays is, you have some assays under development. These are two assays. They are cell transformation assays, and they claim that they have quite a good concordance with the in vivo rodent carcinogenicity assay. And they are able also to detect not only genotox but also non-genotox carcinogens. The problem with this assay, however, is that the mechanisms are not fully understood, so we are kind of dealing with black box, which is not really liked by scientists, and also the subjectivity of scoring. So here the end point is transformed colonies. So someone, an expert, should sit down on the microscope and decide which one are transformed or not, which of course is not so nice, of course. So in vitro, for genotoxicity, we have some regulatory accepted tests like the AMES test and the micronucleus test, which is currently one of the most commonly accepted battery to identify genotoxicants. The claims here are that you have really high concordance with the in vivo assays. However, genotoxicity does not always mean carcinogenicity, and the concordance with the negative in vivo assay is quite low. So actually, they are really sensitive, but also producing a lot of false positives, so that's not good either. In vitro, when it comes to non-genotoxic induced carcinogenicity, there is not much. So the question is, what do we do? And that's kind of the reality, let's say, of non-genotoxic carcinogenicity. Here, actually on this side are the number of, these are non-genotoxic carcinogens, and this is actually information from literature. What are the processes through which these chemicals can induce hepatocellular carcinoma? It's quite diverse, so yeah, it's quite complex. And here we ask the question, what can we do? And we try to use, it's not any more recent concept, but on the adverse outcome pathway, it's going to be developed further in the other lectures, I think, more. But yeah, the adverse pathway is actually in a construct which tries to describe a sequential chain of causal linked events and a different level of biological organization that lead to adverse outcome. And the idea is that all these different key events or molecular initiating event can be targeted by in vitro assay, so they could give some more clarity on how this adverse outcome can be targeted earlier. And the problem, however, with generation of this adverse outcome pathways is that the scientists need to sit down, or a few scientists, and they should spend time to compose those AOPs. Sometimes, of course, this is a subjective process, sometimes incomplete because you cannot follow all the literature. So within the advanced project, our goal was actually to develop a more automated process of data gathering from different databases. So to gather and try to find links between events at different level of organization. So we try to gather information from 4 different databases: ToxCast, CTD, Reactome, and TGGate. And yeah, we actually developed 2 modules. In the first module, we gathered information from ToxCast and CTD and tried to extract relationships between chemicals, genes, and disease. The algorithms which are applied behind are actually using so-called shopping basket approach. So it's trying to bundle all the properties which go most often together. So if you go in the supermarket and you try to buy some Pampers, it's quite often that they are next to the beer shelf, so they would go together. So once we extract these relationships, we try to add some information about the pathways in which they are involved from Reactome. So at the end, we have the chemical gene pathway disease relationship. And we can actually, at this point, filter by any disease of interest to see what kind of information for this disease is available from these databases. This we actually applied for the use case of hepatocellular carcinoma. It's actually an interactive graphic, but it was not possible to. I needed my computer actually to show the graphic, so that's why it's a bit blurred. But at the end, we for hepatocellular carcinoma, we succeeded to derive approximately 240 genes which are involved in hepatocellular carcinoma, and also to group them in all the pathways in which they are involved. But this is a result of combined genotoxic and non-genotoxic exposure, and in order to refine and see which of those genes and pathways are more non-genotoxic specific, we added a second module in the system and extract this based on the information coming from TGGate. We selected a set of reference compounds and used genomics data, histopathological data, and actually the third module is still under development. And we try to extract more customized response. So what we've done, again, for the use case of hepatocellular carcinoma, we selected 10 genotoxic carcinogens, 10 non-genotoxic carcinogens, and 10 non-carcinogens. And in terms of gene expression data, we just did pre-selection according to fold change in p-value and try to extract genes which are present at least in 2 out of the 10 chemicals per toxic class, so to derive more non-genotoxic specific gene signatures. For histopathological data, the workflow is that we focus more on severe and moderate damage, which was detected after exposure to these chemicals, and then we try to select again the non-genotoxic specific histopathology. So once we took the information coming from module 1 with information coming from module 2, we actually with module 2 applied the more specific part on non-genotoxic specific associated genes. So we came out with 5 genes of interest. If you just run a quest through Reactome, these are the official pathways in which these genes are involved, but this is quite general. So what we did is we just go through literature a bit, and what we see indeed what's the relationship, is there a direct relationship with those genes with cancer. And for 4 out of the 5, it was quite good information, and it was they are quite good hit. The first one is involved in double-stranded DNA repair and direct limitogenic in overexpression leading to cancer. Then the second one is oncogene. When overexpressed, promotes proliferation leading to cancer. The third one actually is also quite promising, and when it's overexpressed in tumors, and they are currently trying to promote it as a biomarker for early detection of lung cancer. There's quite nice study on that. And the fourth one is also overexpressed in tumor. Only for ACTA2, there was nothing so substantial as information. We also went into Human Protein Atlas to check a bit more information on those for genes of interest. All of them are cited as cancer-related, and in terms of liver cancer, their high expression is most of the time unfavorable, which means that patients will more probably die faster. So what we did again is just to extract it and try to link the information on those 5 genes with the reactome pathways to see whether it overlaps a bit with information in literature, which of course it did. And we try at the end to try to see where those genes can fit in the reality picture, let's put it this way, which pathways they could potentially, contribute, like to give more clarity. So this is how we try to fit them in. It's not perfect, but maybe that's just additional information needed. The idea also, so what we proceeded further is also with some histopathology. That was information present on TG gates. Actually, it's more microscopy, I put it this way. After exposure to non-genotoxic carcinogens, these are more the specific findings. So you can see more degeneration, proliferation also. The same picture when you, the same information after exposure to genotoxic carcinogens would lead more to necrosis, whereas here it was more, I would say, vascularization, proliferation as a response. So the idea is actually that this workflow we have created is quite automated. So once you can just quest for a disease of interest, and quite fast you can get this information and try to decide how to use it. It's just a faster way of using the information from these databases. And of course, it's not, I'm far away from the claim that this is AOP, but it's just information which might support the development of AOP. Of course, it needs expert judgment afterwards. So that's more or less, I think, from me. It's just, it's more a general conclusion on non-genotoxic induced cancer that maybe, adverse outcome pathway as a concept it's a good way to go, but of course it's just to support its development. This tool is to support its development. And that's acknowledgement to the help I received from the DC members.
Very good, thank you, Tatyana. Any question for Tatyana?